So today begins sort of a shift away from individual new reactions to using what you've learned to understand biology. The theme for the next several chapters is the relationship between biomolecular structure and function. And so we're going to go through many of the classes of biomolecules, starting today with amino acids, peptides, and proteins, and explore this relationship between structure and function, and give you an idea of how changes in the structure of a biomolecule change its function, its shape, its conformation. What we do will set the stage for what you will learn when and if you take biochemistry, Chem 481. I've scheduled 3 days in the syllabus on amino acids, peptides, and proteins. We will probably take longer than that because I just really like this stuff. A lot of my research is in peptide and protein chemistry, and we're going to go a little bit beyond what the book does in terms of structure and function. As part of what we do, you'll be learning the names and one-letter codes and structures of the 20 amino acids, and you'll learn their side chain pKa so that you can make some sensible predictions about the shape and properties of a molecule, of a peptide or a protein. So we'll be a little bit flexible with the syllabus. I'll let you know as we go along in emails about how many more days I intend to spend on this topic, but it's interesting enough, and we'll be flexible. At the end, we'll come back to new chemistry, new reactions with a chapter 28 on some new carbon-carbon bond-forming reactions. But for now, we'll delve into biomolecules. Okay, so most of you have heard of an amino acid before. We'll call these alpha amino acids because they've got an amino group at the alpha carbon relative to a carboxylic acid. So I'm going to draw this amino acid in its neutral form, or actually I'll draw it with the nitrogen positively charged, just so I can show you pKa's. So this is an alpha amino acid. The 20 natural alpha amino acids have L stereochemistry. Now, what does that mean? Well, L and D were the ways of talking about stereochemistry before we had the R and S sort of priority rules. It's not necessarily easy to assign L or D because you do it by analogy to known molecules. So we won't really get into how you assign an amino acid as L or D. I like to remember it this way. When I draw the carboxylic acid group on the right and the ammonium group on the left with the alpha carbon at this sort of valley position, the side chain needs to be a dash. And most of the 20 amino acids, for all but cysteine, the configuration is S. Okay, so just assuming this were some kind of carbon, this would be priority group 1 for the nitrogen because nitrogen's heavier than carbon. 1, 2, 3. Looks like it should be R, but proton is up, so it's S. Cysteine, the amino acid cysteine, is R because its side chain has a sulfur, and so it wins in a priority battle. That doesn't necessarily matter, but all of the L amino acids, all of the 20 amino acids have this stereochemical configuration. The pKa of the ammonium group in an amino acid here is around 9. That's a little bit lower than for an ammonium group, and that's because of the nearby carboxylic acid group, which is withdrawing electron density a little bit away from the ammonium. Similarly, the carboxylic acid group of an amino acid generally has a pKa of around 2. That's a little bit lower than expected for a carboxylic acid. Again, it's inductive electron withdrawal from the nearby nitrogen. For an amino acid, this R group can be any number of things, and we'll talk about that, but in general, we call that the side chain. Whereas this portion of the amino acid which I'll highlight in gray, the alpha carbon, the carbonyl carbon, and the nitrogen we call the backbone. All right, so backbone, side chain. Of course, we'll talk about these amino acids as being parts of larger molecules. So, for example, here, I'm going to draw a tripeptide, and a tripeptide is a peptide composed of amino acids. A peptide is simply a polymer of amino acids. So in this tripeptide, we have sort of 3 different amino acids. One's in the middle, one's on the left, and one's on the right. And I'll draw the carboxylic acid over here as it would be at physiological pH, and I'll draw the ammonium group as it would be at physiological pH. The pKa of the ammonium group is 9, so at pH 7, that proton should still be on. The pKa of the carboxylic acid group is 2, so at physiological pH, that proton should be off. You'll notice that this tripeptide sort of has 2 different ends. One is what we're going to call the carboxy terminus, that is the end of the peptide that has the carboxylate group on it. For short, we'll call this the C terminus. And then over here is the amino terminus, or N terminus. So one of the first things you need to learn about peptides is they have directionality. The amino end is different from the carboxylic acid end. And it doesn't matter how you draw the peptide. You can twist the peptide around, draw it with the amino end on the left side, the carboxy end on the right side. All of that's fine, but you should realize that the 2 ends are different. Okay? And of course, a peptide could be much, much longer than just 3 amino acids. Most of the amino acids in a long peptide chain are going to be in this sort of typical internal environment where the amino acid in the middle is involved in an amide bond with the amino acid to the right and in an amide bond with the amino acid to the left. However, there will be, in any peptide, there will be 2 amino acids in this sort of terminal environment where at the N terminus you've got a positive charge and the C terminus you've got a negative charge. So if you're the side chain at the N terminus, you're right next to a positive charge. If you're a side chain at the C terminus, you're right next to a negative charge. If you're a side chain in the middle, you don't have a charge on either side. A couple other things to learn: the bond between amino acids. You will have learned to recognize that as an amide bond. We also, interchangeable with that, use the term peptide bond. Okay? So in this tripeptide, there are 2 amide bonds. And these amide bonds sort of divide the peptide into its individual subunits. So I'll have a squiggly line here and a squiggly line here that separates amino acid 1 from amino acid 2 from amino acid 3. In other words, this nitrogen, this alpha carbon, and this carbonyl carbon all belong to the same amino acid. And there's an old term, I'm not necessarily sure where it comes from, but any individual amino acid subunit of a peptide is sometimes called a residue. So you'll hear people talk about this particular residue. You wonder, what are they talking about? That's just another way of saying an amino acid inside a peptide or a protein. Okay, questions so far about basic peptide structure, amino acid structure? Okay, good, because it's going to get worse and more complicated. All right, so looking at a tripeptide, there are three interesting bonds that we want to talk about. All right, we want to talk first about the bond between a carbonyl carbon and the adjacent nitrogen. Then we want to talk about the bond between the nitrogen and the adjacent alpha carbon. And then we're going to talk about the bond between the alpha carbon and the carbonyl carbon. And you can see that a peptide is composed of those three bonds in order and then repeating. Amide bond, N to alpha bond, and alpha to C bond. And so we're going to talk about each of those bonds and explore the conformations that a peptide can adopt about each of those bonds. Some of this is going to involve you thinking about Newman projections in ways that you probably thought that you were hoping not to ever do again. Well, sorry folks, we're back. So now I've prepared some slides that are going to help you with this. But you're not going to be able to copy them down. That's okay, because I'll make the slides available to you. And we'll also go to a website that allows you to sort of play around with some of these conformations. Okay, so here is a tripeptide, all right? I've just drawn basically the same thing that I drew for you above, only we're looking at a segment of a tripeptide in an existing larger protein. So this is no longer the N terminus, this is no longer the C terminus. This nitrogen is attached to a carbonyl carbon of the previous amino acid, which we're just not showing. And this carbonyl carbon in green is attached to the nitrogen of a subsequent amino acid, which we're also not showing. That's what the squiggly lines mean. I've labeled the three amino acid residues in orange, purple, and green, respectively. I've labeled the backbone atoms as N, alpha, and C, purple N, alpha, and C, and then green N, alpha, and C. I've also introduced a way of talking about amino acids that are adjacent to each other in sequence. This is something that peptide and protein chemists do all the time. I don't know if you remember learning in math about series where they said, let the ith position be whatever. So we're going to use the term I to refer to any arbitrary position that we're going to start counting from. I plus 1 is one amino acid to the right or towards the C terminus. I plus 2 is two amino acids to the C, towards the C terminal side of this amino acid. So relative to orange, this is the I plus 1 position, this is the I plus 2 position. Of course, you could start counting here and then say this amino acid over here would be I minus 1 and I minus 2 and so on. That's just a way of figuring, just a way of saying I'm two amino acids away from this one. Questions about that? It's not huge, but I think it's maybe sometimes confusing to people. Okay, so I've highlighted around the purple amino acid the three bonds that we're interested in that are going to describe the conformations available to an amino acid in a peptide. The bond between, oops, that's an eraser, the bond between the orange carbonyl carbon and the purple amide nitrogen. That's the C to N bond. Then the bond between purple nitrogen and purple alpha carbon, and then the bond between purple alpha carbon and purple carbonyl carbon. And I've shown these pink arrows describe rotation around those individual bonds because they're sigma bonds. So we're going to talk about what conformations are available there. And each of these conformations is going to have a characteristic dihedral or torsional angle. I'll define that a little bit later, but we're going to use the Greek letters omega and then phi and then psi. And I always remember psi because it looks like Raphael the Ninja Turtle's weapon. I don't know if you guys even know Ninja Turtles anymore. That was a thing of a long time ago. But anyway, omega, phi, and psi are going to describe the conformation available to a peptide by rotation around these individual bonds. We'll use those Greek letters to refer to a specific angle that we'll observe in a Newman projection. Okay, so we're going to use as our example this large protein structure. This is an alpha helix, and in this large protein structure, I have 3 alpha helices wrapped around each other. It's called a coiled coil. We'll learn some more about that a little bit later, not today, but maybe next time. We're zooming in on this particular segment of the coiled coil. This is the orange amino acid at the I position. This is purple I+1 and green I+2, just like we drew it over here. So we're going to zoom in on that, and we're going to look at the conformations we're going to focus on the conformation of the purple amino acid, looking at what these dihedral angles are in an alpha helix. So we begin with the dihedral angle omega. To view the dihedral angle omega, we're basically going to put ourselves in the plane of the page, and we're going to look down this bond between orange carbonyl carbon and purple nitrogen, as we would in a Newman projection, okay? So just imagine what we're going to see. The orange carbonyl carbon is sp2 hybridized. It's trigonal planar, so if we put ourselves in the plane of the page and we look at it, we would see the carbonyl carbon pointing straight up, and then this orange alpha carbon sort of pointing straight down. That's what an sp2 hybridized carbon looks like from the left, from in the plane of the page. Similarly, if we look at this nitrogen, this amide nitrogen, because it's participating in resonance with the adjacent carbonyl carbon, should also be trigonal planar sp2 hybridized. And if we looked at it from the perspective of a Newman projection, we would see a hydrogen pointing down and the alpha carbon pointing up. Here's the view that we could take. I've zoomed in on just these atoms in the structure of this molecule. And here's the carbonyl carbon. In these structures, oxygens are red, nitrogens are blue, hydrogens are white, and then carbons are whatever color I've chosen to highlight. So this is the orange I position amino acid. This is the purple I+1 amino acid. Here's the orange carbonyl carbon. Here's the purple amide nitrogen. With our Anakin Sith eyes, we're looking down the carbon-nitrogen bond. And if we do that, this is what we see. So here is the Newman projection drawn out on paper. The cylinder represents this carbon-nitrogen bond with the carbon in the front. And you can see, we see the carbon-oxygen bond pointing upward, the carbon-alpha carbon bond pointing downward. And then in the back, from this view, we can't hardly see it. We know the nitrogen's there. The alpha carbon is pointing up, eclipsed with the carbonyl oxygen in the front. And then the hydrogen is pointing down, eclipsed with the alpha carbon in the front. Okay? Questions you want to ask about that? Sometimes it takes people a minute to figure out what on earth we're even talking about here. So, yeah. So, right, what is this angle we're looking at? The angle omega is defined as the angle between the 2 alpha carbons. So we're looking at the alpha carbon in the front and the alpha carbon in the back. They're 180 degrees away from each other. So omega equals 180 degrees. The dihedral angle omega is 180 degrees. Now, and that's exactly what you would expect from knowing what an amide bond is, from remembering that in an amide bond, both carbon and nitrogen are sp2 hybridized. So, of course, this alpha carbon would be 180 degrees away from this alpha carbon. That omega angle could have 2 possible values. You could imagine a situation like this, where, as we've said, the omega value is 180. You could also imagine a situation where omega equaled 0. Let me draw for you just briefly what it would look like if omega equaled 0. If omega equaled 0, the nitrogen in the back would rotate such that the alpha carbon in the back was down and the hydrogen was up. And therefore this dihedral angle between the 2 alpha carbons would be 0. Drawn out from the side, what would that look like? This is an amide that has a dihedral angle of omega equals 180. This is an amide drawn out such that the dihedral angle is 0. We call this a trans amide and this a cis amide. In general, trans amides are more stable than cis amides because of sterics. You can see how in the cis amide, the two alpha carbons would start to clash with each other. So the trans amide is more stable than the cis amide. And that cis versus trans amides play an interesting role in peptide and protein chemistry, in part because interchange between cis and trans amides is slow. One reason for that is in order to go from trans amide to cis amide, you have to rotate the nitrogen and you eventually bring the lone pair on the nitrogen out of conjugation with the carbonyl carbon, because at some point along the way from going from trans to cis, you've got to pass through an angle where the nitrogen lone pair is perpendicular to the carbonyl carbon. So sort of interchange between cis and trans amides can be somewhat slow. But in most cases, the preferred orientation is trans. Okay, so omega is kind of easy because there's only two possible values, 0 and 180. Any other questions you want to ask about the omega angle? Okay, let's talk about phi now. Remember, phi is going to be rotation around the purple nitrogen to the purple alpha carbon. Notice that the purple nitrogen is sp2 hybridized. It's only going to have two things attached to it, the carbonyl carbon here and the proton here. In contrast, the alpha carbon is sp3 hybridized, so the alpha carbon is going to have three things attached to it. The alpha carbon will have the side chain, the proton, and the carbonyl carbon. So we will zoom in. Here is a view from the structure. Again, here is the orange amino acid, the purple amino acid, the orange carbonyl carbon, the amide nitrogen, the purple alpha carbon, and the purple carbonyl carbon. We're going to be looking down this bond between purple amide nitrogen and purple alpha carbon. And we're going to be looking specifically at the angle between this bond in gray from carbonyl carbon to nitrogen and this other bond in gray from alpha carbon to carbonyl carbon. So again, our evil Sith eyes looking down this N to alpha bond. This is what we would see if we were just drawing a Newman projection, and then this is what it actually looks like in the structure of the molecule. In the front, we have the purple nitrogen. It's bonded to a proton, and then the carbonyl carbon from the previous amino acid. That's exactly what we'd expect for an sp2 hybridized nitrogen. That's what we see over here. Here's the proton, here's the carbonyl carbon. In the back, we can't see it. It's on the other side of this cylinder that represents the bond between the nitrogen and the alpha carbon. In the back, you've got the alpha carbon, and it's bonded to three things: the proton, the carbonyl carbon, and the side chain. And you can see that here. You can't see the alpha carbon. It's sort of behind the nitrogen. But you do see the bond between alpha carbon and proton. You do see the bond between alpha and carbonyl. And then here's the side chain. Only you can see the atoms of the side chain there. They're not an R group. The angle phi refers to the angle between the two carbonyl carbons. Okay, so this is the angle phi. And in this alpha helix, that angle tends to be minus 57. And that's generally true, around minus 60 for being in an alpha helix. Having that dihedral angle allows a twist that allows the backbone of the peptide to spiral around itself. Now that phi dihedral angle can have a lot of different values, and I'll show you how you can adopt those different values. We're going to go to a website that has a tool that allows us to do that. But some values are more preferred than others. So now we're going to dig deep into our 351 intuition. You remember from 351, you learned that staggered conformations are better than eclipsed. And you learned that in general, you want to keep the largest groups away from each other. Now for that issue of staggered versus eclipsed, you can see the problem we've got having an sp2 carbon bond into an sp3 carbon. When you had two sp3 carbons bonded together in 351, I don't know if you remember ever drawing something like this. If both carbons are sp3, you can draw a perfectly staggered conformation where all the groups are staggered. If one's sp2 though, you see the problem we're in. This carbonyl carbon can be staggered with the nitrogen and the other carbonyl carbon, but now the proton's eclipsed with the side chain. So we have a little bit more difficult time optimizing the conformation when one of the two carbons is sp2 versus sp3. So you got a judgment call. Which is the best conformation? Ideally, you want to keep the two biggest things, which are probably the carbonyl carbons, at least staggered and not eclipsed, but then you've got the side chain to worry about. There are other steric clashes that start to occur in a peptide or a protein, and finding the right conformation is one of the influence of the side chain on which conformation is best is one of the ways that amino acid sequence leads to structure. So I'm going to just go to a website where there is a tool for examining phi and psi angles. There's a link to this in your study guide. The tool is not that great. Oh, it's not bad. Okay. So it's not labeled with color, but we're looking down in green, the bond between the nitrogen of an amino acid and then the alpha carbon. So this, and I don't have a pointer. Let's see. Ah, there we go. So I don't have a laser pointer to use with the Apple Pencil, so I'm just going to have to point up on the screen. This is the nitrogen, this is the alpha carbon, here's the proton, here's the side chain. And the angle phi is going to be the angle between this carbonyl carbon here and that carbonyl carbon there. So right now, phi equals minus 115, that's this angle. And I can change it just by plus or minus, okay? And over time, you're going to see some yellow and red surfaces start to show up, and those indicate places where there are steric clashes. So for example, if I start to adjust phi to be around minus 160, you can see that the carbonyl oxygen is now bumping into the side chain. So that would suggest you want to avoid conformations where phi equals around minus 160 maybe. If I continue to go around, you can see if I'm not careful, as phi starts to get close to 0, I've got a conformation where the two carbonyl oxygens are now bumping into each other. Okay, so there are areas of, there are various angles. Oh, zoom in, that's nice. There are various combinations of phi that generate steric clashes. Now, fortunately, you don't need to worry about all the possible iterations of phi values. You just need to understand the idea that some phi values are going to be better than others. And having a phi value that avoids steric clashes is one of the main ways that amino acids adopt their shapes. Let's go ahead and go to the, oops, we zoomed out. I have no control over the zooming in or out, and I have no idea what I just did. No, oh, that was a terrible idea. I pressed reset, and now it's going to take forever. We'll just wait. Maybe it'll reload. How was the weekend? I took a nap on Saturday, which I don't usually do. We didn't go skiing. Friday night we went to this restaurant in Saratoga Springs called Blossom, pretty good Chinese restaurant. That was fun. Played with my niece and nephew. Oh, there it is, all right. My two nephews and my niece both came over on Saturday. It was awesome. 2-year-old niece wanted me to spin her on something at the park. One of these things that you sit in and then you spin around, and as you try to sit up, you're actually changing your angular momentum so you go around faster. Anyway, she's 2. Do you want to keep going? Yeah. So I kept spinning her around. Are you done? Yeah. I picked her up and set her down, and then I could tell she was going down. And it was funny. Her legs were going, but she was going this way and that way, and I was trying to correct her. It was really, really funny. Okay, so I need to show steric clashes. Yeah, I'm going to take you back around to the phi angle that corresponds to being in an alpha helix, and you can see at least there's no steric clashes. So that's one of the reasons why that dihedral angle is favorable. Okay, so now we'll look down the phi angle, and remember the psi angle rather is now between alpha carbon and carbonyl carbon. So now as we're looking from this direction down that bond, the sp3 atom is going to be in the front, the sp2 atom is going to be in the back. All right, so just scrolling there, here's the view we're taking. Here's the orange amino acid, here's the purple amino acid. We're looking down this bond between alpha carbon and carbonyl carbon. And so the sp3 hybridized alpha carbon in the front, we should see 3 things: the proton, the side chain, and the amide nitrogen. And then at the sp2 carbon in the back, we should see only 2 things: the carbonyl carbon and then the green nitrogen of the next amino acid. So as we look, and here's my drawing, and then here's the view from the actual structure. Here's the carbon in the front, proton, side chain, nitrogen, and then in the back, here's the carbonyl oxygen and the amide nitrogen. And the angle psi is the angle between the 2 nitrogens. In general, the phi, the psi, and the omega are always between the backbone atoms. And here, psi equals around -47. And again, you can see sort of the trade-off, right? I can have the nitrogen staggered, the green nitrogen staggered in between the side chain and the purple nitrogen, but that's going to mean the carbon's eclipsed with the proton. And so you're trying to balance subtle steric issues. And in 351, as I said, you were trained to think about, okay, let's keep the biggest things apart, which is good. But I think you saw in that image that I showed you, there are sometimes steric clashes not only between the atoms that are immediately attached to the bond that we're talking about, but also to other things. For example, there it is. We're just looking at now phi again, but you see the steric clash that we start to see there is not between the carbonyl carbons, but it's between the carbonyl oxygens that are 1 bond out from what we would normally worry about. We'll zoom in on the phi angle, or the psi angle now. This is the angle between the carbonyl, sorry, I'm going to set that to. This is the angle between the the alpha carbon in the front and the carbonyl carbon in the back. And again, we can change that psi angle and sort of start to rotate around. And you'll see again, there are some values of psi where we start to see steric clashes. For example, here, when psi is around 25, we see a steric clash between the carbonyl oxygen in the back and the amide proton in the front. And you can see that if I change phi, some of those steric clashes are going to change. So the idea here is that phi and psi are interdependent. If phi is a certain value, that's going to introduce different steric clashes to psi versus if phi is a different value. So somebody actually went around and tested all of the possible combinations of phi and psi to see where the steric clashes would be, and predicted all of the places, all of the different types of combinations of phi and psi that an amino acid could possibly have. And then somebody else went and measured the actual phi and psi angles for all the amino acids and all the proteins that we had data for, and they match up pretty closely. And what you get when you plot phi and psi together is something that's called the Ramachandran plot, named after the person who first did it. Okay, so in a Ramachandran plot, you show the phi angle along the x-axis, and phi can have values from 0 to minus 180 and all the way to plus 180. At this stage, you're wondering what makes the plus angle positive and the minus angle negative. There is a particular sign convention, and it has to do with the dot product of two vectors and the cosine of an angle. So you don't need to worry about whether phi should be positive or negative. I can't remember what the sign convention is. I think if it's sort of clockwise versus counterclockwise, it's positive or negative. You don't need to worry about it because nobody does the math by hand anymore. What I do when I want to measure a dihedral angle is I open up a program, I click on the atoms, and it tells me what the dihedral angle is. So, but you can see in general that both phi and psi can have values from plus 180 to minus 180. If I were to measure phi and psi for each of the 3 amino acids in this alpha helix that we talked about, we just went through and measured phi and psi for the purple amino acid. Had I done that for the orange and the green amino acids, you would see, here are the data, the phi and psi values for each of those amino acids. And if I plotted them, they all cluster together in the same region of the Ramachandran plot. And in fact, that area of the plot, if you plot all proteins, is highly populated, and proteins that adopt these dihedral angles by definition are alpha helices. So I'll show you an image of the Ramachandran plot from the website that we were just visiting. Each black dot represents one amino acid in a protein. And you can see there are areas of the Ramachandran plot that are highly populated. That means there are lots of examples of amino acids in proteins that adopt that angle. And then you can see there are others where there is hardly anything. And the reason there's hardly anything here, those are exactly the combinations of phi and psi where you see those steric clashes that I illustrated in that program. Amino acids that tend to adopt dihedral angles up here tend to be in beta strands or beta sheets. Amino acids that tend to adopt dihedral angles here tend to be in alpha helices. There is a weird pocket over here of amino acids that adopt exactly the opposite phi-psi angles as you would expect for an alpha helix. We may have something to say about that a little bit later. What I will tell you right now is they call this portion of the Ramachandran plot the right-handed alpha helix conformation, and we may get back to that a little bit later. Okay, questions you want to ask about dihedral angles, phi and psi, and the Ramachandran plot? Okay. I'm going to stand around and wait for a minute until somebody asks something. Because every time I do this, somebody shows up at my office and says, so I have no idea what you're even talking about with the Ramachandran plot. So let's just save ourselves some time and you can ask that question now. Nobody wants to. Okay, the point is that you can be quantitative about describing peptide and protein conformations. Saying that something is an alpha helix is fully equivalent to saying the dihedral angles phi and psi in that peptide are all in this area of the Ramachandran plot, or all have values of around minus 60 and minus 50. The other thing I want you to see is that this is, because we talked about optimizing conformations and keeping stuff out of the way, this is one of the ways that a protein side chain, this R group here, can influence conformation. The size of the side chain can change the sterics around both of the bonds that we're interested in. Yeah?
So is it mostly just the side chains that lead to those slight variations?
Is it mostly the side chains that lead to slight variations? Yeah. It's the sterics of the side chain. It's also the functional groups on the side chain that can sometimes interact with the carbonyl carbon or amide nitrogen of the backbone and make conformations stable that wouldn't otherwise have been. So we'll look at each of the 20 amino acids, and we'll look at what dihedral angles are typically adopted by those amino acids, and you will see there are differences. And one of the key principles in proteins is that amino acid sequence leads to structure and function. That all the information in sequence can be, all the information necessary to specify structure and function is embedded in the sequence. And this is something that now AI has essentially solved, with the Nobel Prize in 2024 being given for AI and machine learning in understanding protein structure. Google's AlphaFold can now, you give it a sequence and it will now tell you what the structure is. And it's pretty dang good at making the prediction. Yeah?
Are we expected to be able to deduce structure from the Ramachandran plot?
Could you deduce structure from the Ramachandran plot? You could know, I would like you to know that this portion is alpha helix and this portion is beta sheet. There's more to it than that, but that's enough for now. Also, understand that I guess if a sensible conclusion it would be to make would be if I said, hey, every amino acid in a peptide has dihedral angles here, it would be a pretty good hypothesis that that's an alpha helix. Yeah, others? Yeah.
So on the plot, the X is the phi and the Y is the psi.
That's right.
What do you do with the omega?
You know, people don't generally plot omega because it can only have two values generally, around 0 and 180. Yeah. What else? Yeah.
What are the main ways we'll need to use this in like a testing situation?
Yes. What are the main ways you could use it on a testing situation? I'll give you an example of that in a little bit when I show you how the Ramachandran plot changes for different amino acids. But in general, I'd give you data and ask you to make a conclusion about the data. Yeah?
Will the data look like it's graphed like this plot, or is it going to be in like numbers?
It could be in this form. I could also provide it sort of tabulated like that. So let me give you an example of this. What we're going to do next is talk about the structures of the amino acids. You should get started now, by the way, on memorizing the structures and names and one-letter codes for the 20 amino acids. And we'll go through each one. But we'll zoom in first on the amino acid alanine with the one-letter code of A. All the amino acids, by the way, share this same basic structure of nitrogen, alpha carbon, carbonyl carbon, alpha proton, and then side chain. So as I draw the 20 amino acids, I'm just repeating their side chains. This is alanine, where the side chain is a methyl group. Here is the Ramachandran plot for alanine in the crystal structures of all proteins that were available at the time. I'll tell you more later about what a crystal structure means. This is just a three-dimensional structure of the conformation of a peptide or protein based on experimental data. So phi on the x-axis, psi on the y-axis. These are heat maps. In a heat map, you use color to indicate population density. So in this heat map, red means a place where there are lots of examples of phi-psi angles of a particular value. And in blue are areas where there's not that many examples. So colder colors means fewer examples, warmer colors means more examples. In this plot, alanine has a ton of examples in what we would call the alpha helical region of the Ramachandran plot, where phi equals around minus 60 and psi equals around minus 50. In contrast, there are fewer examples up here in the region of the Ramachandran plot that's beta. One of the conclusions you could make from this data is alanine's pretty comfortable very comfortable, in fact, very favorable in an alpha helix, and maybe okay in a beta sheet. Yeah? Do you need to memorize the Ramachandran plot? No. Yes. You should know the x-axis is phi and y-axis is psi, and you should be able to interpret a heat map. So if I asked you what conclusion you could draw from this, it would be sensible to say alanine's really comfortable in an alpha helix. Let me show you an amino acid that's less comfortable in an alpha helix. We'll go to, let's see which one. Yeah, let's go to glycine. This is in the category of weird amino acids. You'll notice that glycine is comfortable, it has a lot of examples in the alpha helical region, but it also has a ton of examples over here for positive phi values in ways that the other amino acids don't. I'm just zooming out a little bit so you can look up at alanine. Notice that for alanine, the positive values for phi, there was nothing going on there. But for glycine, suddenly it's fine to have positive values for phi. What do you suppose is going on there? Why would glycine be different from alanine? Here's the structure of glycine. What do you notice about it? There's no side chain. Instead, it's just got 2 alpha protons instead of 1. Suddenly that removes a big steric problem, right? And suddenly that opens up a whole area of the Ramachandran plot that glycine can adopt because it's not trying to avoid steric clashes with the side chain. So this is another dramatic way in which side chain structure influences the conformations a protein or an amino acid can adopt. Let me show you another example of how side chain structure can change the Ramachandran plot. Alanine is a fairly small amino acid side chain. It's just a methyl group. For valine, the side chain is an isopropyl group. The beta carbon, the one that's bonded to the alpha carbon, is a branch point where there's 2 methyl groups coming off. There are a few side chains like this that have branching at the beta carbon, meaning 2 carbons coming off from the beta carbon. We call them beta branched. You'll notice comparing alanine with valine that like alanine, valine's pretty content in an alpha helix. Lots of examples with that dihedral angle. But unlike alanine, valine's highly populated in beta sheets as well. And one of the things you could infer then is adding that beta branching makes this amino acid happier or more favorable, or makes the amino acid better suited for this particular combination of dihedral angles, which happens to be in a beta sheet. So, what could I ask you? I could ask you something like, what does this Ramachandran plot tell you about valine versus alanine in beta sheets? And you could say valine's better in beta sheets than alanine is, and that's a good conclusion. Another interesting thing is if we look at polar amino acids. Let's see, where are they? Sorry, there's too much here. Here is serine. Serine is like alanine but has an OH group on it. You can see that serine, like alanine, is pretty content in an alpha helix. Now, I should have put them side by side. We're going to have to scroll, but we're going to look at another amino acid which is almost exactly like serine, but has a beta branching point. It's going to have a methyl group branching off the beta carbon. If you were to come up with a hypothesis based on what you saw for alanine versus valine, what do you think the beta branching group's going to do to its ability to be in a beta sheet? You predict it maybe is going to increase it. So let's look at threonine where you've got that beta branching, and indeed much redder up here in the beta region. So again, structure related to shape. Structure of the amino acid side chain related to conformation. That's an important conclusion you want to draw. All right, thanks. We'll see you Wednesday.